So, the FDA just approved the first gene therapy drug at the end of last year.
That's amazing.
Yeah, it's called Luxturna.
Luxturna. They always have these weird names. Hi, I'm Emma.
And I'm Rachel.
And we are the Steministas. We educate the public about science and the news. So what is gene therapy? So I know it's come out a lot recently in the news, and we're all about trying to address stuff that's happened in the news.
Mm-hmm. That's actually a great question, because I think gene therapy gets confused with gene editing often. And the big difference is that with therapy, you're delivering a gene to cells, but with editing, you're taking the DNA that's already present in the cell and making cuts in the DNA and making changes. So gene therapy doesn't change your DNA that's there. It's kind of adding another factor.
So it's almost like, if you think of a post office, you're sending an envelope full of a protein or something that you need to your cell, whereas with gene editing, you're cutting out some of the letters that are there.
Exactly. So one of the things you need for gene therapy is a vector. So
like one of those calculus lines that we learned about.
Yeah, there's a vector in every field. In the gene therapy field, a vector simply refers to what delivery strategy you're using. So what are you using to deliver these genes that we're really good at synthesizing in the lab into the actual cells? So in most cases, people are using viruses right now, actually.
So how does a virus deliver a gene to a cell?
I mean, when you think about it, it's really genius, because viruses are literally designed to break into the cell and make more copies of themselves. So they're expert burglars. So before using these viruses as a delivery strategy, scientists remove the parts of the viruses that are pathogenic and that can cause disease, and they simply use it as a strategy to get into the cell.
Okay. Are there any adverse effects that can come from using a virus? Because I know whenever people hear the word virus, they kind of freak out, even if they take out those pathogenic parts.
Yeah, there's always the risk that, these viruses that they use, they're called adeno-associated viruses, or AAV for short. They can be present in the wild. So if you've had an infection with this before, there's a possibility that your immune system could recognize features on the outside of this virus and respond to it in a bad way.
Yeah, no, for sure.
Yeah.
So are you able to design the outside of the virus to look a certain way so your immune system will respond to it well?
Definitely. I think we're still kind of, it's an active area of research, the interaction between these viruses and your immune system, but we're getting a lot better at identifying those features that your immune system can recognize and making sure they're not there.
Okay.
So gene therapy is not an end-all, cure-all, for sure. It's most effective in the cases of disease where you're lacking a gene. So I think a good example is people that are lactose intolerant, right? They don't have this enzyme that can break down lactose. So you could use gene therapy to deliver this enzyme to the cells where they're needed. Whereas if you had a disease where you're making too much of a gene product, like cancer, if you're making lots of oncogenes, maybe delivering more of those oncogenes to the cell wouldn't help anything.
So what would be the benefit of doing gene therapy for someone who's lactose intolerant versus just taking the lactase enzyme with your meal?
Well, lactose intolerant maybe isn't the best example, because with gene therapy, you're not able to have this gene forever, and you may need multiple doses. So for lactose intolerant people, taking the pill would be easier.
Okay. So are people hesitant about gene therapy?
Yeah, definitely. And the field of gene therapy is moving a lot slower than stem cells, for example, part of that is because of a case that happened in 1999 during the first clinical trial for using gene therapy. And this clinical trial was to treat the disease OTCD, where the patients lack this OTC enzyme that helps them break down proteins. So the gene therapy trial was delivering this gene to make this OTC product to the liver cell where they need it.
Okay, so the gene therapy was helping because they were able to then give this person the gene they needed to break down the protein.
Exactly. So, but even without this gene therapy treatment, these patients are able to manage their diet and they can lead a relatively normal life. So It wasn't life-threatening. But one of the patients that was part of the clinical trial, Jesse Gelsinger, was an 18-year-old from Tucson, Arizona, and he had a really severe reaction to the virus that they were using, AAV8.
Wow.
And he actually ended up passing away. And it set the gene therapy field back a lot. They had to completely stop the trial. There's a huge investigation trying to determine what went wrong.
Yeah, so did they ever kind of find out what went wrong? Because scientists take lots of steps to make sure something's safe by testing it in animals first and then maybe going to humans, but like doing a smaller dose. So what happened?
Definitely. So the investigation found that the researchers did a few things, could have handled a few things better. So first of all, when they determined the eligibility for the clinical trial, they didn't take into account the buildup of ammonia, which can happen in these patients. So Jesse had a really high ammonia level.
So does that ammonia come from the virus itself?
No, it comes from the problem that you can't break down the protein properly.
Okay. So it's almost like...
It's like a byproduct.
Yeah.
Even before the clinical trial started, his ammonia levels were higher than the other patients. So maybe the disease had progressed a little too far for him, and someone with a less severe case should have been used instead.
Okay, that makes sense.
So there was a question of eligibility, and
Yeah.
maybe they could have taken into account other measurements than what they considered.
Yeah, because normally they'll screen patients for a lot of different factors, like their age, how severe disease is, if they respond well to other treatments, or what other drugs they're on. And that's normally, that process takes a long time as part of the clinical trial, and is probably the most important part to make sure that you have a well-represented sample of people.
They also found that the team failed to report and be completely transparent about some of the toxicity data with earlier versions of the vector that they used. Apparently they had some primates die in the testing, not with the vector they actually used in these patients, but with earlier versions of the virus.
So they gave it to monkeys, and then the monkeys just ended up dying.
Yeah, exactly. I think they ended up hemorrhaging.
Wow, yeah, it's important to report everything because, yes, it could derail a clinical trial, but in the case of Jesse Gelsinger, knowing that stuff ahead of time could maybe have prevented his death.
Absolutely. The third thing, and something that's going to be a factor in any clinical trial, is that there was a financial conflict of interest. So obviously the researchers and the universities they work for will benefit from this drug passing clinical trials.
If a researcher comes up with a drug and then the university is also a part of that, if the drug goes to market, they're going to receive dividends from that drug going to market. So when it's like something you've invested so much time and effort and energy and money in, and there's a potential for financial gain, I'm sure that motivates them to try and make it happen no matter what.
Yeah, I can definitely introduce a good amount of bias.
Definitely.
So, getting back to the news story that brought me to all of this, Luxturna is a new drug that's being used to treat retinal dystrophy. And this is kind of a broad spectrum of diseases. So they all relate to problems with the retina, which is the back part of your eye where the photoreceptors are present. And these cells transmit the light information to the brain so you can process what you're seeing. So people with retinal dystrophy have defects in these photoreceptors. Maybe they're dying, maybe they're just not functioning properly.
And they'll sometimes have defects in the cone cells, which are the cells that are involved in seeing color.
Mm-hmm.
But then they could also have defects in the rod cells, which are allowing you to see like dark and light. So some people are great during the daytime because they can still see all the colors, but then at night they're basically blind. So there's a wide spectrum of these disorders.
Exactly. So Luxturna is only effective for patients that have a mutation in a specific gene called RPE65, which codes for the retinal pigment epithelium 65 gene. And they have a mutation in this gene.
And this gene's involved in the surface of the retina. So the epithelium is like kind of your skin cells. So within the layers of the retina, there's lots of layers that make it up. So this is involved in that aspect of the retina.
Exactly. So Luxturna is delivered through an injection. You inject the virus into the eye.
So these sort of therapies, how expensive are they for a patient?
Unfortunately, really expensive. Luxturna costs $850,000
For how many treatments?
for one.
Oh my, what?
And sometimes these patients are going to need it in both eyes.
Yeah.
Actually, I think it's $850,000 for both of the eyes.
Okay.
But
I mean, that's still a lot of money.
But still, yeah, and I don't know about how much insurance covers for this.
Wow. But at least with this sort of disease, it's helping make their lives so much better, like a treatment like this.
So now that the first FDA gene therapy drug has been approved, it'll be really exciting to see what comes next. I mean, there's a lot of diseases where I think this kind of technology would be applicable.
Definitely, and I know they're doing more research into like different other vectors for delivery. Like people have done research into the nanoparticles, which apparently don't really interact with your immune system, and you can like coat these nanoparticles with gold, and then they'll just go in and edit the genes. It's crazy.
What we could think of next.
Yeah.